Mutation, the blueprint of genetic variation in bioinformatics. Mutations play a fundamental role in shaping the genetic diversity of life. This educational resource explains what mutations are, their causes, types, and how bioinformatics plays a critical role in their study and real-world applications. Introduction to mutation. A DNA double helix. Every living organism has a biological blueprint called DNA, deoxyribonucleic acid. DNA carries instructions for building and maintaining the body, stored in sequence. However, sometimes changes occur in this sequence. These mutations can be no noticeable effect on the organism, can lead to diseases or genetic disorders, can provide advantages such as resistance to diseases. Mutation equals permanent change in the DNA sequence. Mutations are the foundation of genetic variation, evolution, and adaptation. Causes of mutations. Illustrations of common causes like radiation, chemicals, viruses, and DNA replication errors. Mutations can arise from multiple sources, both internal and external. Spontaneous errors. Mistakes during DNA replication can lead to random mutations. Though rare, they can accumulate over generations. Radiation exposure. High-energy radiation like UV rays and X-rays can break DNA strands or cause chemical modifications. Example: UV-induced skin cancer. Chemical exposure. Mutagenic chemicals, such as those in tobacco smoke or industrial pollutants, can alter DNA bases. Viruses. Certain viruses, like human papillomavirus, HPV, can insert their genetic material into host DNA, disrupting normal gene function. Mutations diagrams, illustrating different types of mutations with labeled DNA sequences. Different types of mutations affect DNA in unique ways. Substitution mutation. One base is swapped for another. If it occurs in a coding region, sickle cell anemia is caused by a single nucleotide substitution. Insertion, deletion mutation. Extra bases are added, insertion, or removed, deletion, leading to a frame shift mutation, which can disrupt the entire gene's coding sequence. Example: cystic fibrosis often results from a deletion of 3 bases. Subsequent mutation, a single nucleotide change in the DNA sequence. Example: it can alter protein function or stop production altogether. Coding versus non-coding regions. Coding mutations directly affect genes and protein synthesis. Non-coding mutations can influence gene regulation, such as enhancer regions that control gene expression. Mutations differ in their effects based on both their type and location in the genome. Mutation and bioinformatic computer screen, showing DNA sequencing data and bioinformatics software. Bioinformatics is a field that uses computational tools and software to study mutations by analyzing large datasets from DNA sequencing. Key methods include genome sequencing, reading the entire DNA sequence of an organism to identify variations. Variant calling, comparing a sample's DNA to a reference genome to detect differences. Mutations. MuteSearch. Real-world applications of mutation analysis. Cancer cells, a genetic report, and personalized medicine. Mutation analysis has numerous practical applications, including identifying driver mutations. Sixth conclusion. DNA, a computer and a microscope combined, representing the fusion of biology, technology, and medical research. Mutations are more than random errors. They are the fundamental drivers of life's complexity. They have shaped the evolution of species, explained the origins of diseases, and paved the way for groundbreaking discoveries in genetics.